Good evening. I would like to thank you all for coming to my presentation. I'm Taylor, and today I'm going to be talking about the effects of decomposition on dissolved oxygen. So to start, what is decomposition? Decomposition is a naturally occurring process that happens in all bodies of water, and this includes lakes, streams, rivers, ponds. If there's water and if there's aquatic vegetation in it, decomposition will occur. And this can be accelerated by many different factors that include nutrients in the water, the abundance of species in a given area, and the weather. So if it's cloudy out, photosynthesis cannot occur as frequently, which will lessen the dissolved oxygen in the water. And if too much decomposition occurs, it can have a negative effect on the ecosystem. And one of these negative effects is that it can deplete dissolved oxygen, which is better known as DO, which I will be saying a lot throughout this presentation. And when decomposition occurs, it consumes the dissolved oxygen supply in the water, and this is done by the bacteria that perform the decomposition. And this can have a negative effect because many aquatic species rely on dissolved oxygen to survive. And one of the most common species that do this is trout, which are very oxygen dependent, so if they do not have enough dissolved oxygen, they cannot thrive in the ecosystem. So the gap in knowledge that I wanted to research further was how decomposition rates among various plant species compare to each other. And this led me to my hypothesis that if surface area to volume ratio is greater, that decomposition will occur at a greater rate. And so this led me to believe that milfoil would decompose quicker due to how dense it was and how many leaflets per growing tip there were, and that Allodia would decompose slower. So what I needed were the plant species, and I used Allodia, Eurasian water milfoil, and coontail, and I selected these species because they are very abundant in Cazenovia Lake, and also Eurasian water milfoil is an invasive species, so I wanted to see how an invasive species compared to a native species when they decompose. I needed beakers to put my plants in, a DO probe to measure the dissolved oxygen, a scale to weigh my plants, and a rake toss to collect my plants from Kaz Lake, which is pictured above. So for the setup of my experiment, I weighed the plants at around 3.9 to 4.25 grams and added equal amounts of water to my beakers. For each of the species, I added 2 of those growing tips that I weighed, which were pretty small, which is why we added 2 to each of my beakers. So in total, I had 20 growing tips per species. So I had 10 400 milliliter beakers per species and control, which equaled 40 beakers for my experiment. I started in the greenhouse, which had adequate sunlight for the plants to perform photosynthesis. Every week I did tests to measure the DO of the water. I did this Monday, Wednesday, and Friday, and this lasted about 6 weeks, so in total I had about 18 measurements. When the DO began to stabilize in the greenhouse, I moved it to the basement and turned the lights off so there was no more sunlight for the plants to perform photosynthesis. I also added distilled water to take into account that evaporation was occurring. And I used distilled water so no extra nutrients were added to the beakers that would affect the DO measurements. When in the basement, I also placed a glass slate over my beakers to prevent diffusion from occurring, which would not mess up my DO measurements further. Like I stated earlier, my experiment lasted about 6 weeks, and if the DO was still changing, I would have done this longer if I needed to. So here are my results. I performed 4 box and whisker pots for my control and 3 species. So I only did 11 select dates out of the 18, and I chose the ones that were the most important. So on September 30th, which is September 19th, which is in the dark blue, is when I started. And then from the orange to light blue was when they were in the greenhouse. And then from October 3rd, which is green, to October 28th was when they were in the basement. So as you can see, the control stayed pretty consistent at about 8 milligrams per liter, which was expected. And then the rest had an increase on September 21st and began to decrease on October 3rd. And then after I did these, I performed a ANOVA test to calculate the p-value. And for all four of my species and control, they had a p-value of less than 0.001, which meant that there was a high significance among them. I also performed t-tests to perform a time-wise comparison, which is indicated by my lettering system. So if they have the same letters, it means that they had high p-values showing no significant difference. And if they had different letters, it meant that they had low p-values showing that there was a significant difference. And for the time-wise comparison, I only chose 4 select dates that I thought were the most important. And this was the beginning measurement, September 21st, September 30th, and then October 3rd, which is in green. And an example of this would be that on September 19th, for control and Allodia, the p-value was 5.96, so I used A to show that there was no significant difference. And then on the same date, Allodia and Eurasian water milfoil had a p-value of 0.003, which showed that there was a significant difference between the 2 species. And I did this for the 4 dates. So as to why this happened was that photosynthesis was occurring in the greenhouse, which is shown by the orange date on September 21st, and this happened for the next 3 times of measurement. And then when I moved them to the greenhouse on October 3rd, decomposition occurred in the basement, which led the DO levels to decrease. And so between my species, there really wasn't much difference. They all decreased at the same time and all increased at around the same time. If I could do this again, I would use an airtight slate instead of the one I used because there was still some room for air to get into my beakers, which could have affected my DO levels. I also would have liked to test the turbidity before and after my project to see how water clarity was affected by decomposition.